Today, I want to be clear that this will be a work in progress presentation. So that's why my email is here, because I just decided to work this today to receive the feedback of people, of members of this society. So if you are watching this in YouTube after the webinar, you have here my email. I will be happy to receive your comments. So I'm going to talk about discordant calcium release in cardiac myocytes, and an overall of the talk is we are going to see discordant calcium release, the definition and background, the causes and relationship with calcium alternans. We are going to look for a discordance index and the consequences of this phenomena, and also the modulation by intervention that affect RYR2 function. So I brought this schematic representation of easy coupling here to be more clear for everyone regarding the discordant process. So we have here three schematic ryanodine receptor clusters and also L-type calcium channels. When the AP comes, there is release of calcium due to calcium-induced calcium release, but not every release units fire at every beat, mainly when frequency goes too fast. So in that case, in the next beat, we can observe that if the previous firing clusters are refractory, the one which was refractory previously can fire and release calcium, and the sequence repeat again and again. So this is discordant calcium release. And I think it is important to mark the difference between two different concepts. One is the synchrony and the other one is discordance. So we have here an example of the synchrony in calcium release, where release starts at different time points along the cell, okay? On the other side, we have an example of an atrial cell, which is the type of cells where discordance has been more in deep study. We have in the middle of the cell higher calcium signal during the calcium transient, but in the next beat we have a low calcium signal here, but more on the ends of the cell. So it's an heterogeneous amplitude of local calcium transients that present the opposite pattern in the consecutive beats. And this also should be related with a phenomena known as calcium alternans, which is a beat-to-beat alternation in the amplitude of whole cell cytosolic calcium transient. We can observe here the amplitude of intracellular calcium transients that changes beat to beat, but this is a very interesting experimental condition where the AP4 waveform was maintained constant. So this demonstrates that calcium alternance can occur without the alternation in the action potential duration or amplitude. I'm not saying this never happens, but it is possible to observe primary calcium alternance. And given the relationship between calcium and voltage, which is known as calcium voltage coupling, the magnitude of calcium release will affect the duration of the AP due to calcium sensitive currents, leading to repolarization gradients, which makes this condition of calcium alternance a highly arrhythmogenic condition in patients. So here I am showing one example of one of the cells I was studying. So this is a recording done in rat ventricular myocytes loaded with fluo4, and this cell was spaced at 5 Hz. So we have in this axis space in microns, and here we have time. We have 1, 2, 3, 4 calcium transients, but if we look at different points of this cell, the amplitude of local calcium release is not the same. But more interesting, on the next bit, the amplitude of the local transient, which was low in the previous transient, is high now, and the opposite in the closed region. So we can obtain patterns like this of local calcium transients. Another thing I would like to separate as concepts is the concept of whole cell calcium transient alternans versus subcellular discordant alternans. We have here one calcium recording. So we have one calcium transient of big degree and the next smaller. And people used to study or characterize this with an index which is called as alternance ratio. We can observe here the difference between the low and the high calcium transient at whole cell level. But here we are back with bidimensional recording of calcium-dependent fluorescence in ventricular myocytes, and we observe how in successive transients we can see the phenomena of discordance having in this part like a chessboard pattern, where there are high, low, and high, and the opposite or the different phase in the closed cellular region. But if we look at the alternance ratio or the whole cell calcium transients, we are not going to see a clear alternance pattern. So we can say this is discordance, pure or almost pure discordance, and there are other conditions where pure concordant alternance can be seen. If we think about the causes of subcellular discordant alternance, two different possibilities have been proposed. One is the preexistence of subcellular heterogeneity in calcium cycling components along the length of the myocytes, like T-tubules, or maybe the expression of different proteins involved in calcium cycling. But also it has been proposed a dynamic pattern forming instability. But I think despite this, there is not a resolution about. I think it's mostly a combination of both structural and dynamic factors. About this, there is one very interesting framework, theoretical framework, which can predict the possibility of having alternance. This group from Los Angeles, James Weiss and Sirin Ku, they proposed the 3R theory, where the random opening, the refractoriness, and the recruitment are key properties of calcium release units that have influence on the calcium alternance occurrence. So random opening is the possibility that one calcium release unit fire a primary spark and is alpha. The refractoriness is a very important property of the calcium release units and calcium cycling, and we are going to explain more in the next slide. And recruitment is the possibility of the occurrence of inter-RYR clusters recruitment deriving from calcium-induced calcium release, but it's not calcium coming from the outside of the cell. In this case, is like what happens with wave-like propagation. Okay, so we can observe that to have alternance, randomness should be in the middle. Also, recruitment should be low or in the middle values, not too high, and refractoriness should be high. If we have more refractoriness, we will have more possibilities to observe calcium alternans. And this is very important because we can observe that also refractoriness have a role in the generation of calcium waves. So with low refractoriness, we have more probability to see calcium waves. That's why some people, or a lot of people, try to reduce the possibility of calcium waves as an antiarrhythmic therapy by increasing the calcium release unit's refractoriness. But what I'm trying to bring here is the potential risk of promoting alternans due to this mechanism, which I think deserves to be studied. So in the next slides, I'm going to show some general characteristics I found when I started recently with the last year with this project. And one is that, again, in rat ventricular myocyte loaded with fluo4, we can increase pacing frequency. We observe here, despite the image is not the best maybe, we can observe more intensity of calcium in this zone without the same intensity here, but more intensity on the sides. But the concept more important for this slide is that it can reverse to normal when we return to normal pacing frequency. Another characteristics which somehow relates with one of the 3R with recruitment is this kind of patterns. We have successive calcium transients, 1, 2, and 3, 4, and we have this zone which is out of phase of the neighboring zones, but due to calcium wave-like propagation or secondary recruitment of these RYR clusters, it becomes synchronized. I don't know if you can observe better in this 3D representation of fluo4 signal how this goes over time. And one consequence of this phenomena is that discordant calcium alternans become unstable, and also whole cell calcium transient alternant becomes unstable as a consequence of this process. Another characteristic related with the occurrence of discordant calcium alternans is that if we look at the whole cell calcium transient, we can obtain some experiments where the peak of calcium is not changing too much, but there are fluctuations or alternation at the end diastolic calcium level, okay? And this is also related with the late recruitment of the non-firing zones by, like also in this image, by the firing, by the early firing zones. In this regard, this phenomena is an extreme situation similar to desynchrony, but in this case, it's not a zone that was late fire, was a non-firing zone that very late, when the rest of the cell is relaxing calcium transient, it fires and delay or cut the diastolic relaxation of the calcium transients. So other very interesting phenomena, at least for me, was to observe that during the calcium transient amplitude frequency relationship, we can obtain this kind of whole cell calcium patterns where we have a negative staircase, but the phenomena underlying this is the occurrence of little zones releasing calcium in a discordant pattern. And somehow, when I was trying to imagine how this is happening, which is the physiology behind all these experiments, I was trying to look at my images, which are not maybe the best experimental approach to see this kind of phenomena. I tried to name as microdiscordance, I was trying to see if this could be possible, and we have here some examples of probably some kind of behavior like microdiscordance. And more recently, I was really impressed about this because in this paper, this group, Dr. Peter Lipp group from Germany, shows that as the frequency, as pacing frequency goes higher, the couplons reliability is lower. So couplons start not being able to respond when the frequency goes high. So it looks like it's possible to correlate microdiscordance with negative relationship at the level of force frequency relationship. That's why I tried to find a proper discordance index to quantify subcellular discordant alternans. Also, we are going to study physiological determinants, the impact of pharmacological modulators of DC coupling, and try to describe the role played by subcellular discordant alternans in the heart. I didn't complete everything of this. This is the end of the project. And for trying to be clear on how we think about this phenomena, I'm going to show the first approach I have followed to measure discordance, but later the results will be coming from the next approach I'm going to show, which is an automatic detection. But we have, again, space and time going down. This is a rat ventricular myocyte based at 1 Hertz and here at 5 Hertz. And we are plotting different cell zones to have one spatial profile of calcium release. We observe the first transient and the second one, and we can see that at 1 Hertz, despite there is not complete homogeneity in the spatial profile of calcium transient, both are quite similar. But at high pacing frequency, like 5 Hertz, we start observing that there are zones that are not able to fire, which were previously firing and are able to fire again, but not in the next transient. So if we do this kind of spatial profiles, we can detect these zones of difference. And if we study the standard deviation of the ratio between these consecutive transients, we can somehow estimate how much discordance is present in the experiment. Next, very recently, we were able to collaborate with Dr. Leandro Somese, also from Argentina, to write a Python-based automatic local calcium transient detection program in which we can split the calcium frames into 2 microns local calcium lines. So we can obtain this kind of local transients profile. And if we reconstruct the calcium transient, it will look like this, being formed by many calcium or local lines, right? So by doing the analysis and calculating the discordance index, I have included only cells showing an alternance, a whole cell alternance ratio lower than 0.1. And that's why when the alternance ratio is higher, it's not point to study a negative staircase because alternance will be clearly apparent. That's what I thought. That's why I am including only this kind of cells. And we have this axis the percentage of amplitude in calcium transient comparing 5 Hertz with 1 Hertz. So this guy is this case is, for example, 90%, and this goes lower, but we can correlate the amplitude of negative staircase with the magnitude of the discordance. But given I wasn't completely happy with this correlation, trying to relate discordance phenomena with a negative staircase, I have followed the advice from Dr. Bill Loach, who is a collaborator in this project, who told me that a good approach to study this is to have two consecutive transients, transient 1 or 2. In fact, the idea in the future is to take more consecutive transients and see which is the best release at every releasing point we have here in the x-axis. So we have here 33 releasing points. So we have here 66 microns of the myocyte. This is the same myocyte at 1 and 5 hertz. And we can say in gray the magnitude of the best transient, which is formed by every best release coming from transient 1 or transient 2. And in red and in blue, we can see real transient 1 and transient 2. So if we calculate the difference between the real transients and the best release transient, we can estimate something I have called, trying to describe it, as discordance-induced calcium release impairment, or DICRI, right? And this is, and I have remembered this early paper published by important researchers in our field that have said that it looks like negative staircase is more likely to be due to a decrease in the amount released at each site within the cell than to a decrease in the number of sites that release calcium. But if we apply the best release approach, we have here 10% of the calculated best release or best transient, and we have the amplitude of real transients at 1 and 5 hertz. And we observe that when more frequency, the DICRI effect goes higher, or the amplitude of calcium transient as a percentage of the best release is lower, right? Again, we are only including here cells that show less than 10% of alternans in the calcium transient amplitude. So overall, in this part of the talk, we can say that whole cell calcium transient patterns can be, there are some patterns that could we relate with discordance, and this try to be a message for people who study the force response frequency in cardiac myocytes, for example. If you see variability in the alternance ratio, or you see variability in tau or end-diastolic calcium alternance, or you see no clear whole cell alternance but negative staircase, we should consider the possibility of discordant calcium release underlying these whole cell calcium patterns. The second part of the talk is going to make focus on the modulation of subcellular discordant alternans by drugs that affect RYR2 function, which are sometimes called as stabilizers or inhibitors. And as a background of these experiments show that decreased ryanodine receptor activity increased variability and duration of systolic calcium transients in ventricular myocytes. Here, Dr. Eisner and Diaz and the group have used tetracaine in myocytes which were paced at 1 hertz, and they show how the transient profile becomes irregular in terms of heterogeneity between different zones, and we have here the discordant phenomena again and also the wave-like propagation characteristic of these intracellular calcium gradients, which are a key feature of subcellular discordant calcium release. So this phenomena makes the transient more, the amplitude more variable, and they make the duration of transient higher. But moving to potentially clinical modulators of RYR2 function, there is a recent work from Dr. Wei Chen lab showing that in a mutant model of suppression of function of renin receptors, if they apply the modulator of RYR2 carvedilol, which is also a beta blocker, in these cells at a high frequency, they can promote, or carvedilol 3 micromolar promote alternans in this mutant suppression of function hearts. So I was wondering what happens when we try to reduce the probability of arrhythmia and calcium waves by using different compounds to stabilize RYR2 function when we pace the cells at high frequency. That's why I have tried by incubating cells, again rat cardiac myocytes, half an hour with 5 micromolar, and in the future the plan is to extend this to lower concentration, more close to the proposed as therapeutic of BK286, which is a non-beta blocking analog of carvedilol, another compound, dantrolene, which is also recognized as RYR2 modulator, and we have increased pacing frequency. We finally record calcium in absence of electrical pacing, just to see if the compounds were effective to reduce calcium waves incidence, and it was significantly reduced by both, so we are sure that they are doing the characteristic effect. And then I was trying to see what happened first at 1 Hz stimulation with BK and dantrolene and in control conditions. We have here the data. We have the amplitude of calcium transients and the discordance index, which is not significantly different, but I brought intentionally this image, this characteristic experiment of BK condition to mark my hesitate about if we are being able to see discordance at this frequency, given that discordance at 1 Hz, at 1 Hertz, sorry, is it's quite different. We have a big heterogeneity in the amplitude of transient between one extreme to the other extreme of the cell. We have here wave-like propagation, but if we compare the amplitude of every local transient, only in this middle region, discordance will be apparent. So I think we need to explore more in depth how to characterize discordance in these conditions, because discordant index wasn't able to detect it. But at 5 Hz, if we compare the incidence of, the magnitude of discordant index in BK and dantrolene, we can see that only BK has significantly increased discordance in the cells, which is, for me, is very interesting, because we would expect that every time we modulate RYR2 to prevent arrhythmia, we are going to promote calcium alternans. But not every compound is the same. Not the mechanism sometimes is not the same. So it's worth to explore these two and maybe more modulators of RYR and see which is the potential alternogenic effect of each. And if we look at the whole cell calcium alternance ratio like this, again we have a significant increase only with VK. So finally, I just want to describe which are the future plans for these projects. One of the plans for this year before lockdown was go to Oslo, try to measure parallel tubule signal and cytosolic, fast cytosolic calcium measurement at the Dr. Loach lab, but we will see after this crisis. And the other plans are to study the possibility of intercellular discordance, which has been reported by, for example, Dr. Wasserstrom group to be present in the heart as a phenomena, to study the relevance of this not tisular, not macroscopic tissue discordance, but intercellular discordance, where maybe some myocytes have an alternance pattern out of phase of the neighborings, and to see how this correlates with negative staircase again. And also in collaboration with Dr. Alejandro Orlovsky from the same institute where I work, we are trying to set this experimental approach to measure intra-SR calcium concentrations in mice injected with AAV9 virus carrying R-sepia-1ER, which will be able to detect the concentrations or the level of calcium in the SR, and to see this kind of profiles, the spatial pattern of SR calcium during discordance, which is one of the big questions I have, and it's also quite, if SR load alternates or not, is a hot topic at the level of calcium alternance. So finally, as a take-home message, I would like to say that discordance is an important factor to consider when studying cardiomyocyte function and excitation contraction coupling modulation. This can be characterized by multiple strategies to study how it's affected in many models of cardiac pathology. And regarding RYR2 modulation, I would like to say, without being conclusive in terms of the results with the number of experiments I have now, it is important to compare the impact of RYR2 modulators beyond their capacity to prevent SR calcium waves. And finally, I would like to thank the people who participate and helped me to start this new line of research for me: Dr. Martin Villa-Petrov, Florencia Racciopi, Dr. Alejandro Orlovsky and group, Dr. Carlos Valverde and Alicia Mattiasi, Drs. Cecilia Mundiña and Matilde Said, Mónica Rando, Lucia Pagola, and Ramiro Martínez Quiroga, and also to the external collaborators, Professor Bill Luch and Dr. Leandro Somese.
Thanks, Luis. We've got questions coming in here, so perhaps I'll, in my chair role, I'll present the questions. Can you see the questions?
Yes, I am going there.
Okay.
Okay. Yes.
So the first two questions touch on a similar subject, and they're from Sina Hadipour Lakmesari and Wahiba Dari, and they both ask, what is the relevance of this cellular calcium alternans to the in vivo situation? Is it just a byproduct of isolating myocytes, or do you also get it in vivo? Okay.
No, because there are at least two papers from Drs. Gary Eistrup and Wasserstrom, where they measure in whole heart confocal calcium measurements, and they observe subcellular discordant calcium alternans at the whole heart level. And it's one of the plans. That's why I want to study what happens in different conditions and with the drugs at the level of intercellular discordance also. We will see intercellular discordance, intracellular discordance, and also the paper with Carveilo was done in whole heart calcium recordings. Yes.
Great, thank you. So the next question is, I think, from Dr. Kumar, which is the phenomenon that you call decree. Can it be influenced by calcium binding to troponin in the sarcomeres?
Well, I don't think so, because I would think that if the phenomena comes from the binding of calcium to one structure like troponin, it will be similar every bit. So what we are observing here is that sometimes one local calcium release part of the cell reaches the best transient amplitude that that part can reach. But I cannot be definitive, but I won't say that is happening. That
I suppose what I would think about is that troponin will change the calcium buffering in the cell, and that therefore may make some of these phenomena you're looking at more or less stable. It might be interesting.
Yes. So you say, for example, in a model mutant or something like that, we could have unstable, higher or lower decree if you have more or less calcium buffering. That's true.
It might be interesting to do your experiments in models of hypertrophic cardiomyopathy, where the thin filament proteins are mutated, and might be interested to see if the phenomenon is different.
Yes, that's right. That would be really interesting.
Okay, your neighbor from Brazil, Rosana Bassani, thanks you for your nice results. She also asks, do you also need calcium overload, perhaps with high external calcium, as well as high rate to produce release discordance?
Okay, no, I don't need to increase calcium in the perfusion solution. We were working at 1 millimolar, but it will be interesting to see what happens with the phenomena at the normal and also higher frequency by changing calcium levels, because remembering the 3R theory, it is possible that higher calcium levels will change some of the properties, like recruitment between RYR clusters, and maybe also if we change calcium load, we are going to change open probability of the Ryan receptor. So it will be another completely different situation to study.
Well, can I then follow up Rosanna's question with one of my own, which is, as you increase the rate of stimulation, you get a marked increase of diastolic calcium. And you don't talk about that, but could this affect the instability and the alternans?
So you say it's not only time the critical factor for the calcium release unit to be refractory, but also is the increase in diastolic calcium. Yes, I didn't think about that, but yes, we should consider that everything is happening at the same time. Not only time is critical. Yeah.
We might inactivate the L-type current locally or something.
Yes.
David, sorry to bother you. Is it possible for you just to increase the amplitude or the sound, maybe? Because when you speak, I don't think we can hear you particularly well at the moment. Is there a chance to just increase your volume, maybe?
I'll do it the old-fashioned way of speaking louder, because
that's fine as well.
Okay, apologies to everybody. Thank you. Wahiba Dari, the next question. What is the percentage of cells showing this pattern of calcium alternans? And would sarcomere reorganization during in vitro culturing explain calcium release binding variability?
Okay, so the percentage of cells showing this pattern was present on the figures. I think it was a bit lower than 50% of cells in control conditions showing some degree of discordance, and higher with BK. And will be sarcomeres reorganization during in vitro. So if you are saying we are not performing these experiments in myocytes after cell culture, we are doing it in freshly isolated cardiac myocytes. But I will expect that if we work with culture myocytes, the patterns will be different, because, for example, after culture cells lose the tubules, so it should be more prevalent discordance due to lack of the tubules.
Great, thanks. So next question is from Delphine Mika. So thank you for your presentation. Is the discordance also observed in other cells, such as sinoatrial node cells?
Okay, I have never seen, I didn't study other cells than ventricular myocytes, but I have never seen that on sinoatrial node cells. I will imagine that cell as very little, but maybe it's deformation. But it's very, very important for atrial, in general, atrial cells. The group of Lothar Blatter have described that in deep, because atrial cells have less tubule density, so that cells as also heart failing cells are more prone to show this kind of patterns.
But you'd be happy to collaborate with anybody with sinoatrial node cells, would you?
Sorry?
You'd be happy to collaborate if somebody gives you sinoatrial node cells?
Yes, of course, yes.
Excellent.
Good. There you go. Delphine, get in touch.
Absolutely.
Let's see.
Next question is from Seth Weinberg, who starts by thanking you for a great talk, and then says, if you perform multiple experiments in the same cell, do you observe the same patterns of subcellular discordance, or does the spatial pattern vary from trial to trial?
So this is a very unstable phenomena. So it has been proposed by a Western group that the nodes, which are the part of the cell where two different zones are out of phase, migrate to the ends of the cells. I didn't reproduce that. I've seen more changes coming from wave-like propagation, but this is very unstable, so the spatial pattern changes if you have new recordings, or these recordings were performed after 1 minute of pacing at each frequency, and after 30 seconds I record. So in some cells I have tried to record in a very late time to see what happens, and sometimes the signal becomes poor, but it's still present, but it is changed.
Okay, thank you. So next question is from an anonymous attendee, which is, do you think mitochondrial calcium could contribute to this discordance?
Yes, I would say yes, because there is a paper from Frank Heinzel lab from Germany showing that the decay of calcium transients can exhibit something like the synchrony. And for that, mitochondrial calcium uptake is very important, they propose. So I will associate that publication with your comment, yes.
But do you think that there are significant fluxes of calcium into and out of the mitochondria on each beat?
I think that's maybe it's a controversial thing, but I think if mitochondria are not homogeneously disposed on the length of the cell, and there is maybe little, but there is calcium uptake, it could change the availability of calcium for the local uptake at the level of SR. But also, I will think that intra-SR calcium diffusion will be a key phenomena here, because that's why I would like to study intra-SR calcium profiles, because I don't know how the diffusion of calcium in the SR conditions the maybe calcium holes in different parts of the SR when the next AP comes.
Great, thank you. So next question from Guillaume Gilbert. Thanks you for this interesting phenomenon in your talk, and asks, can this alternans or discordance be also influenced by the activity of circuit pumps at higher frequencies?
Yes, that will be interesting to explore, maybe in some mice models with some drugs that increase circa uptake. I don't know, but I will think that it is important for the replenishment of SR calcium. That's why maybe first trying intra-SR measurements. I will try also to study the other component of the calcium cycling machinery. Yes.
Great. So Emiliano Diaz, first thanks you for your presentation.
Hi, Emiliano.
Can the stochastic response of ryanodine receptor gating be a potential explanation of discordance?
Yes, and stochastic response of RYR2 is the factor that the mathematical models describe as a random opening of RYR, and it influences alternans, because if alpha or randomness is too high, maybe almost all the RYR will open when the action potential comes and calcium entry from the outside space, and there will be no zones of the cells to be recruited by others and to form discordant patterns. I would say yes.
Has that been simulated? Because I mean, to me, I don't know, if you have 30 or so ryanodine receptors in a cluster, isn't that too many for these stochastic mechanisms to be important?
Well, the Weiss group described that based on the concept of, if you have too high open probability, you will have too many refractory channels. So you won't have a zone to be firing in the next bit. I don't know. Maybe we'll change the pattern of discordance. Yes.
Okay, great. And then Emiliano Medei also liked your talk and asks about how does it influence the results if you work at normal body temperature and at the intact heart level?
Yes. Okay, I have decided to perform these experiments at room temperature because it's the most frequent condition people used to choose in order to have more alternans. It's like a change to have more incidents of alternans and see what happens. But in one paper from also Wasserstrom group in the intact hearts, they refer that there were not big changes. So they have repeat both the experiments, and they have observed discordant alternans at the room temperature and also at physiological temperature. Yes.
So, David, do you mind if I just interject here slightly? Luis, I mean, in 2018, together with James Winter, who has sadly left research for, left academia for a company, a device company. Well, so he did an innovated heart prep on a guinea pig and showed that by obviously pacing at high frequencies, the heart, you can sort of induce alternans,
yes.
Discordant alternans. Obviously, these are not subcellular. These are sort of, on a sort of a whole heart level. But what he then showed is that if you then stimulated the sympathetic, either by directly stimulating the sympathetic nerve or by actually perfusing the heart with noradrenaline, sort of 200 nanomoles of noradrenaline, you could actually suppress these cardiac alternans. Yeah. In other words, when you increase the heart rate, naturally, this is accompanied by an increase in sympathetic response. And this sympathetic response reduces the alternans occurrence. So have you done your experiments in the presence of, isoprenaline, but physiological concentrations, not the ones that will induce arrhythmias themselves, or noradrenaline, and to see whether actually in a healthy heart, provision of some sympathetic stimulus, such as, let's say, noradrenaline, you will still get occurrence of these subcellular alternans, because you expect to see them when you don't have sympathetic response in a healthy heart, but would you see them as well when you have provision of sympathetic stimulus?
I didn't do the experiment yet, but you're right. So it's like a protective mechanism, and it has been, I think, beta stimulation will increase RYR2 function, but also it's going to increase SR load and somehow prevents alternance. I'm not sure about discordance, but I think it prevents both. And the same happens, for example, when, Dr. Eisner have used caffeine. So you can also, in the paper with the suppression of function of ryanodine receptor, on the opposite to carvedilol, they use caffeine. They prevent alternance incidence by reducing the refractoriness of RYR2. I think that's the key, the refractoriness of the calcium release units. But it would be nice to study this phenomena in whole hearts, maybe looking at intercellular discordance and in the presence of beta stimulation. Yes, that would be great.
I suppose my question is, are your conditions physiological, considering you don't have noradrenaline on board when you're increasing the pacing rates?
Yes, are not completely physiological, but maybe are useful to detect if the cells are more prone or less under some conditions like RYR2 modulators or other conditions. But okay, to realize how physiological it is, we could characterize the phenomena in different experimental models.
So you're using it as a model, basically, yeah, but you accept that there are some issues with the model, as we all do.
Yes.
Thank you. Sorry, David.
No, that's great. So the good news, Delphine Mika has come back to you saying she'd be happy to collaborate on synovial node cells. So
okay, great. Yeah, it is worth to present in this webinar.
I tell you, Yildi Euphorus from Santiago, thanks you for your talk. And she wants to know, have you checked to see how the oxidative stress of the isolation could affect the calcium transient, especially by effects on RYR2 and mitochondrial calcium uniporter?
Well, I didn't address that specific question, but the experiment that make me interested on this comes from a paper we published on JMCC about rapid pacing and the effect of rapid pacing in the promotion of apoptosis. So when I was trying to measure after rapid pacing waves, I have recorded the pacing images. So that's why I have observed this phenomena and start reading over the next years. And in that experiment, I have analyzed that all experiments. And in one subgroup, we have used one Ross scavenger, and it looks like without Ross, the cells are more prone to demonstrate discordant alternans. And I thought that maybe oxidation of RYR and also other physiological stimulations of RYR can decrease refractoriness and prevent alternans in the same way you propose that for PKA or beta adrenergic stimulation and also caffeine. That's other component of the talk I was thinking to include, but I have considered the experiments too old to be included. I don't know why.
Great. The next question isn't a question, it's from Bill Louch, who says, great talk. Sorry we didn't manage to get you over to Oslo. We'll manage a visit when the current situation improves. So,
okay, thank you so much for the help and for being available for discussion every time. Yes.
Okay, the next question from Lou Roderick looks a difficult question. Hi, when looking at myocytes with a lower number of coupled RYRs, that's in species such as pigs and humans, the baseline situation involves synchronous release. And he wants to know whether discordance is increased in these kinds of models with increased pacing frequency.
Yes, I'm collaborating with other group from the lab. I wouldn't say the model because it's not my project, but in a model where the loss of tubule is present, we have observed more discordance. So I think I am addressing your question with that. Yes, it promotes more discordance.
Okay, and the last question we've got is again from Yildi Uterus. She says, because you did your experiments at room temperature, there's a chance that endoplasmic reticulum buffering is responding to an ER stress. And what is the nature of calcium buffering in your cells?
I don't know if I understand the question, but I haven't studied how the calcium buffering is in my cell, to be honest. And I don't know how calcium buffering in the sarcoplasmic reticulum could change in my conditions. Maybe you refer that is affected by the temperature. That could be possible, but I'm not completely sure about what happens with intra-SR buffering and discordance. Sorry.
But as far as the normal cytoplasmic buffering is concerned, presumably your cells are normal with perhaps the addition of some calcium indicator?
Ah, right. Yes. Yes. I have included that on my limitations slide I left somewhere. To support that, the only thing I can add is that also, again, in the paper from Wasserstrom group on, I could share the screen, but it will take a lot of time. You can send me an email. They show in a small series of experiments, they show that they change for other dye with a lower calcium affinity to reduce the buffering capacity of the dye, and they say it was very similar, the results. That's why I was quite okay about that. Yes.
Sure, as you say, I'm sure Yildi can send you an email. Right, well, we've had lots of questions, but I think we've reached the end of them now, haven't we? So
I think there's another comment from Yildi.
Yes.
Okay. She's saying that low temperature can activate the unfolded protein response, and some calcium buffering proteins are involved in it. In a normal world, I'd say to you in the questionnaire, this is something you can discuss over coffee. But I think in the COVID world, you have to discuss it over virtual coffee by email or Zoom or something. So
yeah. We can discuss also with Martin Villapetrov, because he and the group Ignacio, they are studying the response, the stress response of the ESR. So they know more than me about that. So they can help me to reply later if you write.
Fantastic. I'm sure it'll keep her happy. Thank you. So thanks to all the questioners, but particularly Luis, thank you very much for a very nice talk, lots of elegant experiments, and thanks so much for sharing what's obviously very recent data with us. I'm sure we all wish you well with this project. So I at least think you're quite good.
Hello to you.
Thank you.
Okay.
Thank you on my behalf as well, Luis. Excellent work, really nice webinar. David, thank you once again for chairing. And we all get the benefit of David as a chair tomorrow again. So join us tomorrow.
Hasta mañana.
See you tomorrow. Hasta mañana.
See you all tomorrow. Bye.
Bye.